Hello and welcome to this tutorial on foRFUN, a tool that aims to facilitate comparative genomic studies between plant species. foRFUN allows 5 different types of input: gene ID, protein sequence, orthogroup ID, batch input, and sequence from an organism not present in its database. We're going to show the typical foRFUN workflow based on the Arabidopsis CCA1 gene, for which we will see its evolutionary history and function. The gene ID usually corresponds to that of phytosome or phylogeny and can be concealed in the Download Genomes tab. foRFUN allows the user to choose between an analysis based on plant organisms or a more general one that incorporates other rhodophyte, glaucophyte, streptophytes, and cryptophyte algae. In this case, we will choose plant. In addition to choosing the model, we see how a series of organisms appear to select which ones to include in the analysis. In this case, we're going to select Ostrococcus, Clepsydridium, Orisa, and Arabidopsis. Finally, we also can select the tree building method between network joining, EPGMA, Bayesian inference, and maximum likelihood, the default. At the bottom of the screen, there are a series of tabs that organize the different analyses supported by the application. It is very important to always include the organism to which the query gene belongs, otherwise an error will be returned. Once the species and model has been selected, the Run button will start the execution. We can see how the application returns a binary tree in which the genes of each species are represented with a different color and the search term is highlighted in red. A text box with the name of the genes is also displayed, as well as a pie plot showing the relative abundance of genes for each species in the orthogroup. All these outputs can be downloaded using the buttons below. The tree corresponds to the gene orthogroup, meaning the set of genes in the selected model species that descend from a common gene. If we want to subset the tree by clades and not by the selecting species, the Collapsible Tree tab allows us to collapse clades to facilitate the visualization of certain areas of the tree. The evolution of this orthogroup can be studied in the Orthogroup Expansion Contraction tab. The result is an interactive plot showing the evolution in the number of genes in the orthogroup inferred for the common ancestors of each clade, marking in red significant gains and in blue significant losses. When hovering over a node, it shows the name of the clade and the number of inferred genes for the current orthogroup in the corresponding ancestor. A gray color means that the orthogroup is not present in the associated species. Once the evolutionary history of the gene of interest has been elucidated, we will enter a different module, whose objective is to analyze the similarity of sequence and domains to see if a candidate gene can have the same function as another known gene. For this, PHylogenomic domains allows a calculation of all the PHylogenomic domains that appear in our protein. Just select the genes we are interested in from the orthogroup and start the analysis. We can see that we obtain a table with the location and description of the identified domains, as well as a representation of them on the protein chains. There are also links to each of the domains to harvest more information about them. The Multiple Sequence Alignment tab allows multiple sequence alignments to be performed on a subset of the genes in the tree to identify conserved and variable parts during the evolution of the species. There are 2 alignment modes. Muscle de novo aligns the selected sequences, while MAFT filters a previously computed alignment performed on the whole dataset, removing all gap columns in the reduced sequence set alignment. The result can be downloaded in a standard FASTA format or in a more aesthetic way by coloring the different amino acids according to their chemical nature. It also returns an interactive alignment, allowing to perform a series of operations like searching for motifs using regex notation. Once the analyses have been used to hypothesize whether 2 genes may have a similar function, the GeoTerms tab allows a functional study to be performed on the orthogroup sequences. As before, the user can select as many genes as desired from the tree and choose a mode of gene ontology terms, biological processes, molecular functions, or cellular components. The analysis returns a list of the terms associated with each gene in the existing annotations, as well as two different plots. The first is an EMA plot, which is used to see if different terms are associated with the same gene. The other is a tree plot presenting a hierarchical organization of the terms based on their semantic similarity. Another type of functional annotation is provided by Keg orthology, again by selecting the genes to be studied, and the tool will return two main results. On the one hand, a table with information on KO terms, which identifies the genes and counts how many times that same KO has been detected in the orthogroup. Also included is information on the metabolic or signaling pathways in which the genes are involved. In this case, we see that CCA1 appears and its involvement in the circadian rhythm pathway. If KEGG pathway results appear, it can be represented by mapping the gene of interest onto them, selecting each of the pathways. We see how our genes of interest are highlighted in red in the image. For both gene ontology and KEGG tabs, links are provided to each term and all outputs can be downloaded as tables or images. Other annotation is literature-based one. This tab performs a direct query to the plant connection tool, which returns biological relationships inferred from plant science articles. It is intended to provide information to supplement gene ontology terms and KEGG paths. In this example, we will enter the search term CCA1 identified in the KO table and the alias search mode, which gives joint information of the search term independently of the names used to refer to it. For more information on the search modes or to refine and extend the result of this tab, we strongly recommend visiting the Plant Connection website with more ways to represent the results. Once the analysis has been performed, the result is presented as a table with two biological terms connected through a type of relationship. To expand or check a specific piece of information, a direct link to the paper from which the information was extracted is also provided. For example, we see that CCA1 of Arabidopsis has a circadian rhythm and the corresponding paper. The final annotation tab allows the exploration of protein-protein interactions based on the string website. We can select one or more genes from a string supported species, and foR will return a table with all the physical interactions of these proteins. It also informs us of the type of evidence that supports the interaction. If it is derived from other organisms, we will talk about an interlog, and otherwise of direct evidence. Another thing that this tab outputs is the orthogroup of all gene targets. In this way, if another orthogroup is overrepresented, as we can see in the pie plot and the corresponding table, we can explore if there has been a coevolution between the two orthogroups to maintain their function through interaction. In this case, from the orthogroup ID provided, we can perform a complete analysis using the orthogroup ID tab. Finally, we can select one or a small subset of genes to create the interaction network. foRFUN provides an image of the network, and it also queries a string API to create a custom network. We can access it and use the rest of the string tools to get more information, for example, three-dimensional descriptors or smart domains when available. Once we have shown the different information that foRFUN provides, we will move on to the different inputs it supports. If we cannot determine which is the ID of our genes, we can use the Sequence tab to search for it. In this way, we paste the protein sequence, select the organism from the database, and foRFUN will return the same results as in the gene ID search, with an additional table showing the most similar proteins in our organisms. The tree and the rest of the results are referred to the first protein that appears in the table. The orthogroup ID-based search is designed to explore the results from the string tab of other searches. It works identically to gene ID search. Different is the new organism search. In this case, we based one sequence, but it does not have to correspond to one of the organisms in the database. We will use the CCA protein of Elianthus annus as an example. foRFUN first finds the most similar orthogroup based in sequence profiles, and then places the new gene in the correct place of the tree, allowing subsequent analysis of the orthogroup. Finally, the batch mode enables the bulk information retrieval for a list of either gene IDs or sequences from one species in the dataset. We must upload the file with one ID in each line or in FESTA format for sequences. Select the organism, the model, and the search type. PharaohFUN outputs an interactive heatmap showing gene abundance in the selected species for each query's orthogroup and a compressed folder. The folder contains a list of the genes and their corresponding orthogroups, and in subfolders for each query gene, the individual information of most of the tabs. In this way, it returns the gene tree, the expansion contraction tree, a table with the genes in the orthogroup for the rest of selected species, the gene ontology table, the KO table, and the multiple sequence alignment in FASTA format. If the sequence search has been selected, it also returns the identification table with a format similar to that of a blast search. All the proteins used in the construction of the tool are available for download in FASTA format in the Download Genomes tab. In the Whole Dataset tab, the complete orthologs table can be subset to download all the orthology relationships between species of interest. We hope this tutorial has been helpful. For any questions or suggestions, please contact us via GitHub or email.